few words about me. I spent my career at the intersection of science, innovation, and business execution, primarily within the life science services and CDMO industry, and most recently as the Chief Scientific Officer of Catalent, where I led global R&D team developing products, technologies, and services for customers, while also advising the company on entry into new modalities such as targeted protein degrader and cell and gene therapy. So during my time at Catalent, I had the privilege to being part of the John Chiminski's team and help build one of the largest CDMO in the world. And this provided me with a unique hands-on experience on how CDMO became critically important to the life science ecosystem, something that went very, very clear during the COVID period. So equally important, I think, is my experience with also all central CDMO all to the life science innovation ecosystems, which is something important for GPM. So because CDMO work and we work with cross modalities, mechanism of actions, and seen hundreds of programs, Catalent in a given year at 1,000 molecules under development, and we gain a unique vantage points on emerging science. And it's not just only about breadth, it's let us detect which innovation have the potential to be truly transformative if you got really that curiosity and you really want to make a perfect job for the innovator. So many examples stand out, but one that remained very, very vivid to me is Richard Miller and Brurica, a program where those early insights are translated into a game-changing therapy. And now I'm even more excited to see that it's bringing the same spirit of innovation in its ITK program at Corvus, right? But more recently, I've transitioned into board and advisory role, working with life science investors and innovators, which give me a front row view of how science, capital, and execution are being recalibrated in today's environment, right? And that context, I work with investor, innovator, and CDMO to help them define where to focus, how to differentiate platform and capability, and how to align strategy with execution. So, and one thing that I focus upon is, as I believe in the resilience of founders under the current world. When I develop a conviction in a team, such as Prolific Machine, who's going to present on the Wednesday sessions at GPM, I partner very closely with them to set directions and strategy and execute with discipline, bringing the kind of getting things done mentality and mindset we develop at Catalent so that we turn ambition into market impact and at scale. Yeah. AI, yeah, sure. That topic got me back to the bench of the university over the past 2 years, right? I had to get a grasp of it and as a scientist, understand what it could do to our world. So everyone is talking about AI, but I think the more important question now is exactly the one you ask. Where are we seeing real, tangible return on investment? So starting with discovery, I do support organization in that field too. It's fair to say that AI is now embedded across most pharma discovery engines. So there's at the same time excitement and skepticisms around when we'll see AI design drug routinely entering the clinics. My view is that we should remain patient. Biological validation still takes time. But that said, I'm hearing from others and company I'm working with that meaningful clinical moves and data will come this year, and those will be important milestones. But where I see much clearer and nearer term return on investment is in development. Right, drug development is fundamentally an interdisciplinary, data-driven process. So I've been living that world since the past 26 years. So design small experiments, you generate body of knowledge needed to deliver safe, efficient, and manufacturable medicines. And scientists spend an enormous amount of effort and energy trying to identify trends, root cause, deviations in order to bring products and processes under control. So in that context, there is tremendous opportunity for AI to augment scientific judgment. And John Chiminski says in his own word, calls it to assist scientists. And I don't think the answer is a single prescriptive platform, whether you got custom solutions, integrated system. I believe that AI applied to development can realistically save months, not weeks per program. And that is a real economic value where you got returns. So, and if you extend that lens to biomanufacturing, things become even more compelling and interesting. So today, bioprocessing technologies, cell line development, upstream, downstream purifications are really mature. the titer are plateauing, right? You seen the burst that we had in the early 2000s in titer. Now it's kind of plateauing around the 10, 12 gram per liter for the best case maps, and even struggling below the gram per liter for some of the more complex beasts to express. So, but unlike small molecular or oligonucleotide, we're dealing with living systems with inherent variability and limited direct control over it. And the industry has traditionally relied on those indirect approach, selecting clones that balance growth and expressions, optimizing cultural conditions, and to quote a very good friend of mine who retired from Catalan, Greg Black, essentially keeping the cell happy so that they perform. So no matter how much automation or analytic you layer on top, there remains a fundamental loss of control of patient controls at the biological level. So this is why my interactions in a couple of years ago with Denis Kant, the CEO and founder of Prolific Machine, really started. Denis applied optogenetics in non-pharma bioprocessing at the time, using light-controlled genetic switches to directly, dynamically, and reversibly control cellular mechanism and a wide variety of cellular mechanism. It's like putting a button to click in on and off the cell machinery, but also tune it. So when you combine that with AI-driven closed-loop optimization, you suddenly gain direct control over biology rather than getting an indirect observations and doing everything that you could around it. So I believe, therefore, from the early days that this could represent a new standard for bioprocessing, addressing some major unmet needs such as productivity, expression of complex constructs, intractable ones, and also significantly reduced footprint. So congrats to Denis and his team that made the bold decision to pivot fully into biopharma. The early results you will talk about at GPM on Wednesday afternoon have validated that hypothesis. In less than a year, using non-optimized integration method and low copy number, they exceeded 12 gram per liters on a complex map with clear line of sight. I believe they're going to go above the 20 gram per liter on the fed batch system. They successfully express complex therapeutic protein that all other CDMO fail to express, and even viral vector using a triple transfection process and keeping the triple transfection process with optogenetics in it that many CDMOs struggle with. And they recently demonstrated the technology at a 200-liter scales using a self-optimized AI system. So to me, is really what real return on investment look like for AI, right? Putting in application with the right technologies. So therefore, AI in pharma won't be measured by ambition, but by execution. And at GPM, I be attentive to who is actually delivering that kind of impact and consistently and at scale.